Welcome to Azad Bioacademy. Today I will talk about bacterial cell wall synthesis and associated antibiotics. Okay, so I will try to explain in molecular level. That means from the building block to the final cell wall synthesis. Okay, so let's go to the detail. First of all, I will try to give you some basics of bacterial cell wall. You know, the bacterial cell wall is wall that means it is situated just outside of the cell plasma cell membrane basically, and the cell wall, the basic component of the cell wall is peptidoglycan. So peptidoglycan, that means peptide and glycan, that means sugar and some amino acid assembling form is responsible for the formation of peptidoglycan, and peptidoglycan is the basic unit of bacterial cell wall. Okay, so how the peptides will be assembled within the cell and how it will exit from inside to outside, and how the sugar part will be associated and how it will contribute for the synthesis of bacterial cell wall. Okay, so come to the point. I will just try to explain in three categories. First of all, that is initial stage, or you can say initiation. That is basically cytoplasmic stage. Cytoplasmic stage. Okay, that means all the associated procedure or all those associated process will be within the cytoplasm. The second one is, you can say, elongation and transmigration, or you can say transport simply, and that is the membrane stage. Okay, and third one is extracellular. Third one is basically, it is peptidoglycan synthesis stage, or you can say cross-linking stage, and according to the location it is extracellular stage. Okay, so one after another I will try to explain. So firstly, I will go for initiation, that is cytoplasmic stage. What will happen inside the bacterial cell to produce the building block of the peptidoglycan, basically all of the bacterial cell wall. Okay, so if you think this is, you can think like a wall which is basically built up of bricks and cement and sand. Okay, so such type of materials will be very necessary for the formation of bacterial cell wall. So come to the initial stage, that is first one, that is initiation, or you can say initial stage, or as it is happening within the cell cytoplasm. So this is the cytoplasmic stage. Okay, so come how and where it will produce. Okay, firstly, I'll say glucose is the basic precursor molecule to produce the sugar part. Actually, just initially, just few minutes earlier, I said the sugar, that is glycan, and the peptide chain, that is amino acid assembling form. So two types of sugar is the basic molecule to produce the basic skeleton of sugar chain. That is N-acetylmuramic acid and N-acetylglucosamine. Okay, this is the two basic sugar. and some amino acids that will form peptide, that is pentapeptide. I will go further. So that is glucose, or you can take also fructose, and it will follow some steps, and then it will turn into UDP, N-acetyl glucosamine. Okay, that means UDP N-acetyl glucosamine comes from glucose. Okay, so here from several steps. So I will not go for that. So now, already I said N-acetyl glucosamine and N-acetyl muramic acid, two types of sugar, are very essential. So now, one enzyme will work on that molecule, and that will turn into NAM. So you can say that is mur A B, or another name is enol pyruvate transferase. Okay, that enzyme will work on it, and it will turn into UDP N-acetyl muramic acid. Okay, that means N-acetyl glucosamine will be converted into N-acetyl glucosamine, which will convert it into N-acetyl muramic acid by the help of the enzyme. Okay, so this enzyme is very much important. Some antibiotic will work on it. I will just explain in antibiotic stage. The next, that N-acetyl muramic acid will carry some amino acid, that is peptide. How the peptide will be added to the NAM sugar unit, then I will explain now. So now, one enzyme that is MUR C, that will add a peptide or amino acid actually to the NAM sugar. So now I will go to the structurally, I will try to explain. So now here is, you can think, this is a sugar unit, that is, you can say NAM, and one UDP is attached here, UDP. So this is UDP, okay, uridyl diphosphate. And one amino acid will attach to the N-acetyl muramic acid by the help of MUR-C enzyme. And then I will just draw like one amino acid. This amino acid name is L-alanine. L-alanine will attach to the UDP N-acetyl muramic acid. Okay. Now the another enzyme will work on it, that is the MUR-D, and that will add another amino acid to form a dipeptide, that is the N-acetyl muramic acid, and this is already attached in the UDP, and here one is the L-alanine, and now another amino acid will attach here by the help of MUR-D enzyme, that is D-glutamine. Okay, So now again another amino acid will, another enzyme will work, that is mur E, and it will add another amino acid to the dipeptide chain, which is attached with the N-acetyl muramic acid. Okay, so here the sugar part, and this is UDP is attached here, and one is your ALA, that means alanine. Then this is D-glutamine, and now third amino acid will attach here, that is L-lysine. Okay, L-lysine will attach with this dipeptide. That means now this UDP N-acetyl muramic acid is just carrying 3 amino acid, okay, as a just trimer of 3 amino acids: L-alanine, D-glutamine, and L-lysine. Okay, now another enzyme will work, that is the mur F, and this amino acid will attach 2 amino acid dimer at a time, and that amino acid dimer will form firstly. So I will show you, that is here the sugar unit, that is the N-acetyl muramic acid, and UDP is attached. Then here one is ALA, then D-glutamine, then L-lysine, okay, L-lysine, and by the help of mur F, it will add dipeptide, that is one is here, another one is, that is basically D-alanine and D-alanine. That is dimer form will attach here. And who will form dimer actually? In our human body, alanine is available there, but for bacterial cell, D-alanine is very much important. So L-alanine will turn into D-alanine, and then D-alanine will form a dimer, and then it will attach by the mur F. So here I will just try to show you in a short. That is, firstly you can take, that is L-alanine, okay. Then on enzyme will work here, that is racemase, and it will turn into D-alanine, okay. And another enzyme will work here, that is that is ligase, okay. Then what will happen? Another D-alanine will attach here with that D-alanine. So D-alanine attach will And this D-alanine also come from the same way. So D-alanine and D-alanine, it will produce D-ala and D-ala. Okay, so that will attach here by the help of this amino acid. Okay, so this is the one basic unit or molecule, basic molecule, which will contribute to form peptidic glycan. So this is the UDP N-acetylmuramic acid, which is just now carrying pentapeptide. So L-alanine, D-glutamine, L-lysine, then D-alanine, D-alanine dimer. It's just carrying pentapeptide. So some species of bacteria can carry 4 amino acid, 3 amino acid, but most of the cases pentapeptides are more common. Okay, so this is happening within the cytoplasm. Okay, so this cytoplasmic stage, we can say this is the initial stage, or you can say cytoplasmic stage, because this is happening in cytoplasm. Okay, so this is the cytoplasmic stage. So just I will try to just sum up by just keeping some basic things. That is, glucose will be converted fructose, then fructose on phosphate, then it will turn into UDP N-acetyl glucosamine. This is one of the basic unit, and another unit is the N-acetyl muramic acid. And N-acetyl glucosamine will turn into glucosamine by the help of enzyme mu-AB, or you can say the another name is enol pyruvate transferase. Okay, then it will produce UDP N-acetylmuramic acid. Then 5 different amino acids will attached by different enzymes. Okay, and everything is happening within the cytoplasm. So this is the initial stage, and this is the cytoplasmic stage. Okay, so now come to the next stage, that is second stage, that is membrane stage. Another name is called elongation and transmigration stage, or simply you can say elongation and transfer stage. So for easy understanding, I will draw here the membrane. That is, that is the lipid bilayer, and this is the bacterial plasma membrane. And this is called membrane stage because membrane will carry different types of enzyme, and that will contribute to convert the sugar that already we got inside the cytoplasm, that is N-acetyl, UDP, N-acetyl muramic acid, just which was carrying pentapeptide. Okay, so that will convert into the another form, and it will be associated with another sugar. Okay, so now what will be happening here? Now I will just try to explain. So in the membrane, it will carry specific types of lipid, and that lipid, carrying initially 1-phosphate. Okay, so this lipid name is called bactophenol. Bactophenol. You can say also C55P. That is, the carbon 55 number of carbon containing lipid, and which is carrying 1-phosphate. So C55P. You can say simply lipid P. Okay, lipid phosphate. Or in another chemical name is caprynyl phosphate. Okay, so different name. But for your easy understanding, I will several times I will try to use the terminology bactophenol. Okay, so now this is the bactophenol, and that bactophenol initially it is carrying on phosphate. Okay, so this is the bacterial monophosphate containing bacterial. That bacterial, and here another enzyme is there. You can think this is another enzyme. This is called MRAY. Okay, and this on the function of the enzyme is to attach the N-acetyl muronic acid containing pentapeptide containing N-acetyl muronic acid to that bacterial, because bacterial is very important lipid to just carry that the building block of the peptidoglycan, and finally it will help to flip up the basic unit from inside to outside. So come here. So that will add that, the monomer previously formed unit was UDP, that was UDP, N-acetyl muronic acid carrying pentapeptide. Already I mentioned by different color, that is, L-alanine, this is D-glutamine, this is L-lysine, and this is the dimer of D-alanine and D-alanine. Okay. So, in the earlier stage, if you just recall, then you will get the exact figure. And that will attach with that bacterial by the help of enzyme MRAY. Okay, how it will attach? That is here, UDP here, UDP. So that UDP, that means uridine diphosphate. So what will happen? Uridine 1-phosphate will flip out from here and 1-phosphate will attach here. So you can add the phosphate from here and it will be attached to the sugar. Okay, so finally what will happen? That stage I will show you here. So this is the bacterial, and this is the diphosphate, and that is the sugar unit. That is the N-acetyl muronic acid, and that is the pentapeptide. Okay, pentapeptide. So now this pentapeptide containing sugar is attached with the bacterial by diphosphate. Okay, so this is called lipid 1. Okay, lipid 1 means that pentapeptide containing N-acetyl muronic acid is completely attached with the bacterial by 2-phosphate. Initially, it was carrying the bacterial carrying 1-phosphate, and now it is in that form. Okay, and now another enzyme available there. That enzyme name is MORG. Okay, the function of that enzyme, that enzyme will just add another sugar. Already, if you just recall, that was N-acetyl glucosamine. Okay, so then that N-acetyl glucosamine will add here by the help of that enzyme. So now you can add bacterial. This is diphosphate. This is your muramic acid, and here another sugar will add, that is the N-acetyl glucosamine by the glycosidic bond, and that muramic acid will carry the previously attached pentapeptide. Okay, pentapeptide. Actually, this is the lipid tube. Actually, this is the building block of peptide glycan. This is actually the building block of peptide glycan. But some species of bacteria also carry pentaglycine. That means additional 5 number of glycine just attached with the third number of amino acid, that is L-lysine. Okay, so here you can consider the special type of bacteria, that is, you can say Staphylococcus aureus, that will carry pentaglycine. But if you think E. coli, it does not have any pentaglycine. Okay, so I will explain in the later one what is the function of pentaglycine, and without pentaglycine how it will form peptide glycan. So now some enzyme will work here, that is FEMX, FEMA, FEMB. That enzyme, the function is to add glycine, that is single glycine will be added by that enzyme, double glycine, and then double glycine will be added where? It will add pentaglycine here. So if you just add single, that is one glycine by X, then double glycine by A, FEMA, then by B, another 2 glycine. That is the pentaglycine. Okay. So in a sense, some species of bacteria, you can say this is the building block of peptide glycan. Okay. Now what will happen? Now another enzyme which is present in the membrane, that is a single enzyme. Another enzyme is actually 2 enzymes, but those enzymes work together and function like a single enzyme. The name flippage enzyme, that basic unit will transfer from inside to outside. That flippage enzyme main function. So what will happen here? The basic unit containing the bacterial, it will completely exit from inside to outside. That means it will cross membrane through by the help of flippage enzyme. It is flippage. Okay. So now you can draw here outside. So that would be this is your bacterial. This is containing one phosphate and another phosphate, and now it will be attached here like this is the N-acetyl muramic acid, and then this is the N-acetyl glucosamine, and it will carry pentapeptide. One is Ella, second one is D-glutamine, third one is L-lysine, and then D-alanine and D-alanine. Okay, so this is the building block or basic unit of the peptidoglycan. Now what will happen? This is phosphate and the bacterioprenol. The number of bacterioprenol is very much limited within the bacteria, so it is very much important for the bacteria of the membrane. Actually, the membrane enzyme, the main function is very crucial to just flip out the basic unit. So what will happen? The number of bacterioprenol is very limited, so it will be recycled. How it will be recycled? It will be recycled by an enzyme that is called phosphatase. Phosphatase. You know the function of phosphatase, that means it will dephosphorylate. Okay, so from here, phosphate will just out, and one phosphate carrying the bacterioprenol will again embedded to the initial stage, embedded to the membrane. Okay, so now it will free, that means it will be like that. So here you can now draw here, like that is the bacterioprenol, and it is carrying single phosphate, and then it will attach here again. Okay, so you can omit this phosphate here. So this is the unit of building block unit. Actually, the pure, that is the complete unit of the peptidoglycan, that is cell wall, like a brick, no cement, no sand, that is a brick. Okay, so now we will discuss the third step, that is the extracellular step. So I will just sum up very quickly. That means within the cytoplasm, the N-acetyl muramic acid containing pentapeptide will attach the bacterioprenol by the help of MRY enzyme, and then it will form a lipid 1, and lipid 1 will convert it into lipid 2 by the attachment of another sugar, that is N-acetyl glucosamine, and that will form the lipid 2, and that lipid 2 is the basically building block, but some species of bacteria will also carry the pentaglycine, and that pentaglycine containing unit will flip out by the help of enzyme flipase, and that is the enzyme, that this enzyme name is MURJ, and this one is FTSW. The two enzyme combinedly the flipase activity. Okay, and here the lysine containing pentaglycine. Oh, sorry. Okay, so this is all about the membrane stage, or elongation and transfer stage. Okay, so remember the flipase enzyme basically MURJ and FTSW. Okay, so I will just now come to the third stage. That means extracellular stage, or peptidoglycan synthesis stage, or you can say cross-linking stage. You can consider this is the membrane, that is lipid bilayer, okay, and everything now will be happening outside of the membrane, that is within the periplasmic space, okay. So you can think you already got a sugar, that is N-acetyl muramic acid, and which is attached with the N-acetyl glucosamine, the basic chain of the sugar chain, and you know that N-acetyl muramic acid will carry pentapeptide. First one is L-alanine, second one is D-glutamine, third one is, that is L-lysine, and fourth and fifth one are basically D-alanine and D-alanine. Okay, so this is the one unit. So another unit will come up by the flippase or bacterial. So that unit will attach here. That unit will attach here. So you can add here. So this is the N-acetyl muramic acid, and this is the N-acetyl glucosamine. Okay. So now another unit, that is N-acetyl glucosamine, then N-acetyl muramic acid, and N-acetyl glucosamine and N-acetyl muramic acid. We'll add here by the forming bond here, that is the glycosidic bond. Okay, this is the glycosidic bond, and that bond will form by the particular enzyme. This name is that is PBP, penicillin binding protein. You can also it is also known as transpeptidase. Okay, and the transpeptidase or penicillin binding protein has several types. Okay, basically it is two types. One is high molecular weight PBP and low molecular weight PBP. High molecular weight PBP has class A and class B. So class A has two functions. One is transpeptidation, that is cross-linking, and another one is transglucosylation. Okay, that means it will form a glucosidic bond. Okay, this class A, and class B has one single function, that is cross-linking. So both class A and class B is responsible for causing cross-linking, but class A is also another specific function, that is glucoside bond formation. Okay, that means transglucosylation. And the low molecular weight PBP, that is transpeptidase enzyme, has some role, that is decarboxylation. You can say endopeptidase activity. That means it is basically responsible for remodeling or maintenance of the peptidoglycan layer. Okay, so it has some decarboxylation, that is cleavage activity, and embedding activity, that is remodeling activity of the peptidoglycan. Okay, so you can say remodeling and maintenance activity. So this is okay. So now both high molecular weight and low molecular weight combinedly work together. That means combinedly work to form a cross-linking and peptide glycan formation. Basically, peptide glycan formation, cross-linking formation, both enzyme works. So it has multiple number of transpeptidase activity, but in a single bacterium, that is single species, almost 6, 7, 8 transpeptidase enzyme will be available to form the peptidoglycan layer. Okay, so now here one PBP, that is transpeptidase enzyme, will help to form a bond between two N-acetyl glucosamine and N-acetyl muramic acid, glucosamine and N-acetyl muramic acid. And if you think it is happening here outside of the membrane, so where the enzyme will be there? Actually, this enzyme, the penicillin binding protein, that is transpeptidase enzyme, is basically attached to the membrane. Okay, and it will work from here to there. That means it has connection. But actually, for your easy understanding, I'm drawing here out just far away from the membrane, but it is very adjacent to the membrane, and it is, you can think like it is like that enzyme there. And if it is work here, the enzyme is there. Okay, it is attached to the membrane for your kind information. So now this unit will also carry the pentapeptide. Actually, I'm sorry, I forgot to add the pentaglycine. So for your easy understanding, I'm drawing here. That is the pentaglycine. Okay. So here it will also carry the pentaglycine. Okay. So now what will happen? Another unit, that is the basic unit of sugar chain, that is skeletal unit of sugar chain, that will carry the peptide, pentapeptide. And what will happen? Another layer will be there and it will make a cross link. So what will happen? Now you can draw here. This is just I'm drawing here. This is, you can say, now this is NAM, this is NAM, and there that is alanine, then glutamine, then and finally it will also carry pentaglycine. Okay, in that way, again another unit of sugar, and then another unit of sugar, and here will be there also here also it will carry the penta sugar. So now just I am simply I will now try to make you clear about the cross linking. So here the enzyme will work. That enzyme is transpeptidase. That transpeptidase will work on the fifth number of 4 and fifth number of amino acid, that is D-alanine dimer. Okay, this is the D-alanine dimer, this is the D-alanine dimer, and this is the D-alanine dimer. The structure of D-alanine dimer, between the D-alanine, carbonyl group is there. Okay, so this is carbonyl group there, and transpeptidase enzyme in the active functioning position, that active position of the enzyme will carry serine, and serine has OH group. That means if you think this is the enzyme, and here serine are there, and serine will carry the OH group, and here the carbonyl group. So now that enzyme will attach here. The enzyme will attach here. This is the enzyme will attach here. This enzyme is PBP, penicillin binding, that is transferase enzyme. And initially it will attract to each other by the hydrogen bond. Invisible hydrogen bond will attract to each other, and then it will form a bond between them. That is, you can say, acyl enzyme intermediate. So it will form a bond there by acylation between them, and it will form a acyl enzyme intermediate. Then what will happen? When it will form a acyl bond with the dipeptide, then what will happen? Sorry, the bond stage will be there. Okay, so then what will happen? Here, it is basically the high molecular weight PBP, and it is also attached with the low molecular weight PBP. That means combinedly it is working, that is transpeptidase. So what will happen now after binding with the dipeptide D-alanine, and it will form a acyl bond, and then the low molecular weight penicillin binding protein will cleave that fifth number of D-alanine. It will cut off, that means it will cleave. And by the cleavage of that fifth D-alanine, here energy will produce, and that energy will make that bond very strong. That is, covalently it will form esterification, that is acyl ester bond with the fourth D-alanine. And here that fifth D-alanine is just gone. Okay, and energy will produce, and that energy will work to form this bond very strong, and it will facilitate to work with another pentapeptide. Then what will happen? It will just manage, that enzyme will manage to form a bond by the pentaglycine, will make a crosslink. That is glycine, 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 and glycine. So the bond will be between 3 to 4 linkage. Number 3 and 4. One chain, another chain will be attached by the cross-linking. Okay, that is pentaglycine cross-linking. And you know the pentaglycine is Staphylococcus aureus carrying pentaglycine. So between them it will form a cross-link. So here also the pentaglycine will form with the 4th link. So in that way it will form a bond by pentaglycine. Okay, so which species, some species of bacteria like E. coli has no pentaglycine. So in that case, what will happen? In that case, actually, that will happen like, actually those are very adjacent to each other, and it will form a direct bond between amino group of that third number of carbon, amino acid, and here carbonyl group, and it will form a bond, covalent bond, that is amide bond. Amide covalent bond will form, and it will attach directly by 3-4 linkages. Most of the bacteria, the bond is available between 3 to 4. Some bacteria 4 to 3, some bacteria 2 to 4 may be possible, but most of the cases 3 to 4 linkages. So two types of cross-linking, actually I am trying to focus on that two types of cross-linking. One is 3 to 4 cross-linking by pentaglycine, and 3 to 4 direct cross-linking by covalent bond, basically amide bond, you can say peptide bond. Okay, so this is the cross-linking by the enzyme help of transpeptidase, or you can also say penicillin binding protein. Okay, so this is all about the third phase of the peptidoglycan. So you can consider this is the peptidoglycan layer, and you can consider it the cell wall, because the cell wall, the basic component of the cell wall is peptidoglycan. So this is the peptidoglycan synthesis or cell wall synthesis. So it has 3 stages. One is your cytoplasmic stage, that is initiation. Second one is the membrane stage, that is elongation and transmigration. Third one is your extracellular stage or peptidoglycan synthesis, or you can say cross-linking stage. Now I will explain the antibiotics which are basically associated with the cell wall synthesis. So what types of antibiotics are basically cell wall synthesis related antibiotics or associated antibiotics? Now I will explain. Okay. So firstly, in the cytoplasmic stage, I will go firstly for cytoplasmic stage. So in cytoplasmic stage, you know, one thing that is the formation of NAG to NAM, that is N-acetylglucosamine to N-acetylmuriatic acid. Of course, initially UDP attached there, and here you know the enzyme name is mu-A, or you can say also mu-B. Alternative name is enol pyruvate transferase. Okay, that enzyme will be inhibited by phosphomycin. Sorry, phosphomycin. So that enzyme will be inhibited by phosphomycin. When that enzyme will be inhibited, so no NAG, that is N-acetyl glucosamine, will be converted to N-acetyl muramic acid. You know, both the sugar are the basic unit of building a block of the sugar chain, basic skeleton of sugar chain. Already you know. In the third phase, already I tried to show you that is N, A, M, and NAG linkage by the glycoside bond. Okay, so when that enzyme will be inhibited, so NAM, that is N-acetylmuramic acid sugar, will not be produced. So further procedure will not be possible. Okay, so without basic unit of the NAM and NAG with pentaglycine, how is it possible to produce bacterial cell wall? So it is indirectly associated with bacterial cell wall synthesis. So this is one within the cytoplasm. Cytoplasm, single stage. So A, you can say the one. So B is, you know, in bacteria, D-alanine, the use of D-alanine is very prominent than human. So D-alanine. So now L-alanine should be converted to D-alanine. D-alanine. If you recall, the fourth and fifth peptide, that is amino acid of the pentapeptide, were D-alanine. So this is D-alanine, and thus L-alanine will be converted to D-alanine by the help of enzyme racemase. Already I said in the first initial stage, okay? So racemase, and that racemase enzyme and D-alanine will be attached with another D-alanine, that is D-alanine to D-alanine by the help of enzyme ligase. Okay, both enzymes will be inhibited by cycloserine. You can say D-cycloserine. Okay, D-cycloserine. So, how is it possible to form the basic unit or the basic building block without DLR, fourth and fifth number of amino acid of pentapeptide, D-alanine, D-alanine? Without D-alanine, the linkage is not possible, because basically linkage will happen between 3 and 4. So fourth number D-alanine. So without D-alanine, bacterial cell wall is not possible. So the cycloserine antibiotics will inhibit the enzyme, this one and this one. And this is happening within the cytoplasmic stage. So this is the antibiotic which will work in the cytoplasmic stage by the inhibiting some enzyme. So now within the membrane stage. In membrane stage, now it is, if you just recall the membrane, so you will get two things. One is bacterioprenol. So bacterioprenol, what is the function of bacterioprenol? Bacterioprenol will carry the basic unit and it will form the lipid 1, and then it will form the lipid 2, and then lipid 2 will flip out with the bacterioprenol. So without bacterioprenol, how is it possible to attach here so initially it is 1 phosphate is there. So without this, how your another phosphate and sugar, that is glucosamine, sorry, N-acetylmuramic will be there. And you know the N-acetylmuramic acid will carry the peptide. So is it possible without bacterioprenol? And bacterioprenol is inhibited by an enzyme that is called vancomycin. Vancomycin. So vancomycin is just attached with the bacterioprenol, and bacterioprenol function will be inhibited to form lipid 1, lipid 2, and go to the outside of the membrane. So it's not possible because vancomycin will just attach with the bacterioprenol. So this is the function of or mechanism of vancomycin on bacterioprenol. Another thing just recall, that is when bacterioprenol with the lipid 2, that is your sugar, N-acetylmuramic acid, N-acetylglucosamine with bacterioprenol and phosphate, diphosphate will be flip out. That was like that. This is the bacterioprenol. This was your diphosphate, phosphate and phosphate, and that was your sugar, that is N-acetylmuramic acid, and that was your N-acetylglucosamine. And their pentapeptide, including pentaglycine. Okay, so now another enzyme, if you think, that was your that was phosphatase. That's phosphatase actually. This phosphatase enzyme will be inhibited by an enzyme. That will inhibit the dephosphorylation, and it will inhibit the embedding of the bacterioprenol to the membrane. Bacterioprenol to the membrane. So who will do that? That is your bacitracin. That bacitracin antibiotic will inhibit to recycle the bacterioprenol to the membrane. So without bacterioprenol, already I said it's not possible. So bacitracin is basically working to inhibit the recycling, and vancomycin or to inhibit the function of the bacterioprenol. So this is the antibiotics, which are basically working in associated membrane. So membrane associated antibiotics are vancomycin and bacitracin. Okay, and now we will discuss the third phase, that is your peptidoglycan synthesis stage or cell synthesis stage, cross-linking. So this is the very much important, that means in the third phase, those are the beta-lactam antibiotics. This is very familiar, So beta-lactam antibiotics are basically, penicillin, cephalosporin, monobactam, carbapenem. Those are the beta-lactam antibiotics. Why it is called beta-lactam antibiotics? All the antibiotics carrying beta-lactam ring. Beta-lactam like ring, beta-lactam ring. It will just for that structural matter, these are the beta-lactam antibiotics. So all the beta-lactam antibiotics will work on penicillin binding protein. That is transpeptidase enzyme. So now I will show you how it will work. So here it will work if you just recall the sugar chain, and that sugar will carry pentaglycine for your understanding. So this is the D-alanine and D-alanine. And D-alanine D-alanine dimer structure is very identical with the beta-lactam link. Okay, beta-lactam link. Very identical. And then what will happen? You know, this is the important point of the transpeptidase enzyme to attract here and to form a bond, because here D-alanine will maintain, or just you can say manage the all criteria to form acylation, esterification with the enzyme. Because here, if you think this is your enzyme EBP, that is transpeptidase, and that will be said in, already I said during the transpeptidase formation or cross-linking. So now the basic things is, so this enzyme will bind here to make a bond to just cleavage the fifth one, and it will make energy, and that will facilitate to make a cross link between the another pentapeptide. Okay, already I said. So now what will happen? The beta-lactam antibiotics, identical with that structure. Okay, so now what will happen? This beta-lactam antibiotics will bind with their beta-lactam antibiotic will bind with the transpeptidase enzyme. Transpeptidase, that is BBP. Then what will happen? This enzyme will be blocked, will be inhibited, and no chance to bind with the 4,5 dipeptide of D-alanine. It's not possible because already blocked by the beta-lactam antibiotics. Then is it possible to cleavage the fifth D-alanine? It is possible to produce energy. It will be possible to form a cross-link between two pentapeptides? Not possible. So without cross-linking, your membrane your cell wall and peptidoglycan will be more porous, will be more looser. Then what will happen? The peptidoglycan or cell wall, the main function of cell wall is to maintain the osmolarity. Another function is the protection, but one of the major functions is to maintain the osmolarity. So what will happen? When the cross-linking will not be possible, then it will be very loosened and osmolarity will change, and lots of water will enter into the cell and cell will be burst out. Okay, so this is the mechanism of action of beta-lactam antibiotics. That is, you can say here it will bind penicillin, it will bind cephalosporin, it will bind beta-lactam, and so on. So you can say as like the competition to bind here. Who will bind here first? And okay, competition is actually between here to there and here to there. So when it will bind first there, so it has no chance to bind there, and without binding here, not possible to cross-link. So this is all about the mechanism or the antibiotics associated with the cell wall synthesis. So in the initial stage, membrane stage, and outer membrane, that is the peptidoglycan or cell wall stage, different types of antibiotics working. So this is all about. Thank you very much.